Great. Thank you everybody for joining. We're really excited to have y'all. We have a great presentation from Karl Pfleger today of Aging Bio Angels, and he's going to talk to you about investing in aging and longevity today. Paul, I'd like you to come up as well. If anybody has any questions in the audience, you can throw those in the chat box or raise your hand, anything like that. We'll get to you. Yeah, also, we'll have a poll at the end of the session for you about Rejuvenation Technologies, who you'll also hear from. Great company. So feel free to pull that as well. And with that, Hall, I'm going to have you come up and introduce Karl.
Hey, great. Thanks, Shane. And thank you guys for joining today. And we welcome Karl Pfleger. He's an angel investor in the longevity space through a group called AgingBiotech. You can find him at AgingBiotech.info. And let's have Karl introduce himself. And also, Karl, if you want to go and get your slides ready, love to have you go and kick into your presentation. But tell us more about yourself and your work there.
All right. Hi. Can everybody hear me okay?
Yes, we can.
All right, great. So that was a pretty cool little video intro there. Where are my slides? So I'm Karl Pfleger. I am well, I had a different window up, but that's okay. All my slides can be found here at Aging Biotech Overview 2023. I'm actually a tech background guy, but I've been working in the aging field for about 10 years now. I'm an angel investor in startups in the field, specifically in the aging and longevity subcorner of biotech. And I also run a website called AgingBiotech.info, which is a great way to follow along what's happening in the aging and longevity sector. And so that is going to be a good portion of what I talk about here. So as I said, I'm an angel investor. So if you want to see my disclosures, sort of what companies I've invested in, you can find them on my LinkedIn profile, which you can find here. At this URL where you can find the slides, the links are clickable. And later on, there's some papers, and those are going to be clickable too. So first of all, I just want to emphasize that the most important four things to remember about the aging and longevity sector are, first and foremost, the geroscience hypothesis, which is the basic underlying theme of the whole sector. It's that biological, meaning molecular level changes, are what constitute aging, and those are the causes of all top killer chronic diseases. So, cardiovascular disease, cancer, all the neurodegenerative diseases, diabetes, etc. And the other three most important things are that aging is mutable. Decades of recent science show that we can interfere with the process, slow it, possibly halt it, or even reverse it. And doing so is very worthy. It's from a sort of effective altruism point of view, it's one of the best ways to improve the world. And it's sort of a moral imperative, given the number of deaths. Over 100,000 people a day die basically due to aging. And also, it's going to be lucrative. A lot of people think this is going to transform society, that we're at a moment a lot like the beginning of the web and smartphone revolution, and that this is going to be big. Okay, so why should you listen to me? So I run this website that is the broadest and well-respected overview of the field. And I'm one of the most prolific investors in the field. So this is a table on the website showing repeat investors in multiple companies in the space, and this is my column. There's a mix of both VCs and angels in this list, and there's some of them that are reasonably prolific. My Ph.D. from Stanford's in the data science field, originally AI and machine learning within computer science, but I've now been working on the aging field for about a decade. So this is what I'm going to talk about. I'm going to try to make this 20 minutes and then do as much Q&A as people want. We're going to talk about the field and how to use this AgingBiotech.info website to basically get an overview of what's going on and follow along, and a broad overview of what ways to think about how to break down the field. And then we're going to talk about investing in biotech startups in the field. And sort of those two bullets are the the sort of, promise of tomorrow stuff. And then we'll just spend 5 minutes quickly talking about a few interesting things to keep in mind when looking at a topic that a lot of people are very interested in, which is sort of what can you do today with current lifestyle and current clinical medicine. So AgingBiotech.info is a website that tries to collect sort of all the important things in the field in one place and link to them so that you can find more information, get up-to-date info to make sure that you have the latest info. So primarily it grew out of my investing work originally, where it contained a list of all of the companies that I could find. And my list of companies was much, much greater than everybody else's on the web. And that is the way it's launched in 2019, and that stayed that way for a couple of years. And since then, it has expanded to be sort of a one-stop shop for all the best books, podcasts, blogs, the conferences to go to, for researchers, a bunch of free databases that you can get access to, what discussion forums you can use, what journals to read, et cetera, et cetera, et cetera. It also has a one-page summary of all the best counterarguments to objections to working in the field, like overpopulation. The basis of the site is that it's a purely pro bono, non-profit, non-commercial tool. So there's no, it's not selling anything. It's asynchronously updated, unlike a lot of the articles you'll find about trying to give you an overview of the space, which sort of come out at a particular date and then gradually become out of date. And it's focused on content, not flashiness, but it's got a lot of functionality for sorting and restricting. And it's only public info. So this does not contain any secret information from conversations I have as an investor or anything I hear at a conference in a side conversation. Everyone can be sure that this info is already out there in the public, and they can usually verify it by going to directly to the source. So the biggest table is this list of over 300 different companies in the space in the companies table. Right now, I'm a little behind on updating the companies table. So there's over 170 or something like that on the main list. And there's a to be considered sub-tab there that has another 100 plus more that need to be moved over, some of which are just at the moment small, so not as urgent. Like everything on the site, it's not just a list and it's not just a bunch of prose with hard to read text. Instead, everything's organized into tables and lots of different fields or columns for every different entry or row. In this case, every row is a company, and there's loads of different columns. So showing there's a short description of what it does. There's a description of whether it's a diagnostic or a therapeutic, what clinical phase trial it's in, how many people work for the company, whether it's public or privately traded right now, what geography it's in, and things like what modality it uses. Is it small molecules? Is it biologics? Is it gene therapy? Links to the papers. And at the right side of all the columns, there's an as of date. So you can tell, self-documenting in the site, how out of date anything is. And most of the columns, most of the cells are links so that you can click on the information and and find out for yourself more details and or how out of date anything is. So obviously in a 10 minutes or 15 minutes, we don't have time to go into what 300 different companies are doing. But it's important to just understand that a lot of this is super interesting work. It's worth it if you're interested in this field to spend a little time just perusing some of these companies and what they do, and clicking on the links and clicking on their pipeline document and checking out almost every one of them has a page on their site talking about the science underlying the company. And there's a lot more going on in academia, and it's impossible to get a full overview of all the thousands of labs working on aging and biotech-related biology across the world. But the great thing about focusing on the company as the fundamental unit to follow is that anything that's going to get into thousands or millions of people and actually help them is going to be brought to market via a company. So these are the guys that are on the forefront. Okay, so let's talk about how do we think about the whole field sort of top down. Highest level is very easy, right? Just like normal clinical medicine, there's diagnostics and there's therapeutics. Within the therapeutics, roughly in my estimation, about 2/3 of therapeutics are using a mechanism of action whose basic idea is to slow the rate of aging. We know from lots of science on model species and lots of human epidemiological data that one can affect one's rate of aging. It's not a clock that ticks at a constant rate like an atomic clock. And a lot of the companies are just trying to slow it down by 1% to 30% or something like that. About 1/3 of the field is really focused on, let's try to actually reverse aging. Let's try to do something that jumps us from age 65 to age 55 or 60 or 40 if we can eventually. And there's even controversy in the field about whether that's possible, but a large fraction of people definitely think that's possible, and quite a few of the companies in the field and a lot of the papers are working on that goal. On a more fine-grained level, there are a couple different ways to break down the biology of what's happening and what is aging and how can we interfere with it. A lot of people take a divide and conquer strategy, and 20 years ago there was a effort to categorize all the different types of damage by the SENS Research Foundation, and they came up with 7 different kinds of things that go wrong with aging that they think each one of which can be individually fixed. 10 years ago, there was a very famous paper that's become the dominant view in the field called the hallmarks of aging. And then right after that came out a different one called the pillars of aging. And that one kind of lost the marketing war and the hallmarks dominated. And then just recently, the hallmarks have been updated to add a few more. And so there's 7 or 9 or 12 or 14, depending on how you look at it. If you smush them all together, because they all kind of have some overlap, but each one of them has things that the others don't have. So I actually combined them all and smushed them together and figured out which ones were the same and which ones weren't. I think this is the first public mapping. Most of the academic papers don't actually acknowledge the others when they look at one or the other of these paradigms. But roughly speaking, I think there's about 17 or 18 different categories that it's reasonable to categorize companies into in terms of what they're working on. And so there's a category column in the company's table, and this is the sort of mapping of which hallmarks and things it goes into. So we don't have time in this talk to go through even each one of these sort of 12 or 15 or 18, however many you want to do it. But there's a bunch of obvious ones, right? So some of the people are working on mitochondria, some on stem cells, some on proper protein folding. Each one of these things has aspects that go wrong in aging. Some on extracellular matrix degradation and advanced glycation end product cross-link formation, some on telomeres, etc. Each one of these could be a whole 20-minute or 30-minute talk by itself. We don't have time for that, but let me just throw up one of the areas is cellular senescence. Big hot topic in the field. Some cells enter the state where they secrete chronic inflammatory signaling, and those seem to be pro-aging, and getting rid of these kinds of cells in mice extends the lifespan of the mice by 20 or 30% and makes them healthier. Those are all extra healthy years. So there's over a dozen companies working on various therapeutics, and you can get a summary like this for any one of these areas in the site by just sorting by the category column and then restricting. And so this table was generated by looking at just the ones that are working on cellular senescence. And they have many different possible mechanisms of action. So there's a whole bunch of them that are working on just killing those cells via many different means, activating the immune system, using gene therapy, non-viral gene therapy, using prodrugs, etc. So, I don't have time to go into every single thing about each one of these senescence companies, but I urge you to pick whatever area you think is interesting and look at what all the different modes of action. Mitochondria is another one where there's so many different routes that companies are trying to fight mitochondrial aging. It's very interesting and exciting right now. So I'm just going to spend 2 slides or 3 slides talking about the growth of the field. This is a field that's growing very rapidly. The inherent efficiency of going after the things that happen in humans in the 40s and 50s that underlie all the chronic diseases we get in the 60s, 70s, 80s is going to cause it to take over all of clinical medicine eventually, or all of the chronic disease portion of clinical medicine, which is the top killers. And it's going to grow to be from a small corner of biotech to be a very, very large fraction of biotech, possibly eclipsing oncology. And this is the current growth rate from its small base. We have a steady growth from the beginning of the pandemic on, despite the problems with the pandemic, both in terms of number of companies going from 150 to 300, 350, and total employees going from a couple thousand to almost 6,000 now. We can track the clinical trial stages, and the companies that were at a certain stage 2.5, 3 years ago are, have gone, for the most part, there've been some failures, but of course, but for the most part, they've advanced in their clinical stages. One of my favorite quirky ways to measure the growth of the field is just look at the number of companies with names with the roots age, longev, or rejuve or juve in them, and you get this clear cut from about the year 2010, where it was hardly anything. You get this clear cut up into the right hockey stick. So it's a growing field, and I think AgingBiotech.info is a great way to follow along. So now we're going to move on to how to think about investing in early stage aging and longevity biotech companies. And mostly this is how to think about investing in any biotech company. My particular emphasis is that I'm an angel, so I invest in seed and Series A, and and pre-seed friends and families, and bridges between seed and Series A, and occasionally bridges after Series A before Series B. I do invest in diagnostics. Not everybody in the space does, but within therapeutics, I tend to shun the slow rate of aging kind of companies. I don't invest in calorie restriction mimetics or rapalogs, but there are many companies that can be put into the category of slowing aging, and it would still be a huge win for the economy. One paper that came out suggested slowing aging by 1 year, and everybody would add $37 trillion to the GDP. But I tend to focus on reversing aging. So if you want to find other investors in the field who do a lot of deals in the space, go see the Investors Cross Companies table, and those are good people to network with and compare diligence with. These are sort of the decision drivers when evaluating a startup, and how I value them. Some people invest primarily in team. I don't. For me, the most important thing is the mechanism of action. I want to know what the science is and whether it's a reasonable goal. Obviously, if it's at an early stage company stage, then the companies will not have proved the case for that MOA being right or their delivery mechanism being right. There's obviously going to be some risk, but I want to make sure that I invest in things where the MOA is reasonable, where it's hitting a reasonable thing that's really involved in aging, that's fundamental. And if you get something that's fundamentally involved in aging, then you'll have a lot of good shots on goal for good indications. Sorry, I touched the mouse the wrong way. Okay, so early data is very important. IP is somewhat important, but not a big deal to me. But obviously, different people have different emphases about these. The why now important question is, of course, always very important. And a lot of times, it's good to check out the papers. And oftentimes, there are relevant papers, preclinical. So the important thing about aging and longevity as a subsector of biotech that makes it different from conventional pharma drug companies is that any startup is inherently risky and has a low chance of success. So that risk part is sort of similar to every other fields. And the startup investing bet, of course, is always this roulette wheel thing. You have a big payout, but a small chance of that payout actually happening. With aging biotech, because of the fundamental nature of the geroscience hypothesis and the fact that the molecular changes underlie so many different chronic diseases, the calculus is a little different. You still have an epsilon chance of success, but instead of your reward being this pot of gold, you actually have many pots of gold if you win. So if it's really a fundamental aspect of aging that you're fixing, then your pot of gold will have a much easier time label expanding to many other indications or being prescribed off label. And so that's the reason why I think this area will be significantly more lucrative than other areas. All right, so that's what we're going to talk about in terms of investing. So who's asleep? I can't see many in the audience. I don't even know if there's anybody here, but sleep is super important. So let's just spend a few minutes talking about what is actually there's a lot of interest right now in what's actually available today to extend lifespan and keep people healthy. And there's a lot of data on that. And of course, there's a lot of controversy. And so there are people who are taking the early science from the aging longevity field and jumping on board, like using the diabetes drug metformin, even as a 30-year-old non-diabetic, or taking rapamycin because it's the thing that's most reliably extends lifespan in mice. So it's good to be skeptical in this area. And I want to just throw a couple thoughts in front of everybody. Of course, sleep is super important, and sleep, diet, and exercise are sort of the foundations of this, as well as, of course, not smoking and not abusing alcohol or other substances. So how do we think critically about these things? The most important thing is that dose is key, and that subject cohorts and populations and how they're drawn matter, and controls matter. And mechanism of action is important to understand. There's a couple sacred cows, like human data is always better, and randomized controlled trials are the gold standard, but that actually isn't a good. I won't go so far as to say it's not true, but those are actually overdone a lot. RCTs aren't actually the best in a lot of ways, and human data is not always better because of the inherent limitations. So let's go over a couple examples. Metformin is this diabetes drug that people started taking notice of because diabetics on it were outliving demographics match controls who weren't diabetic, despite the diabetes should shorten lifespan. Let me just also emphasize that if you go to the slides and pull them up on your computer, these paper squares are all clickable. But who were the controls in these experiments that made people gaga over metformin's ability to affect lifespan? If we look at the epidemiological data, there's lots of papers that show that healthy lifestyle makes about a 10 to 15 year difference. There's a more recent one where it's a 24-year difference. I don't know if it went up because of the opioid epidemic, because they added not having opioid addiction to the list from these earlier papers. But, so we've got 10 or 15 or maybe 20 plus years difference from, doing all the right lifestyle things. So does metformin really just reposition us within that range? It's not clear yet. But I would be skeptical about taking, for example, someone who's overweight and diabetic and generalizing that to somebody who's lean and non-diabetic and young. Rapamycin. It affects the mTOR pathway, which is super central to metabolic rate of aging. And rapamycin inhibits mTOR and extends lifespan reliably in mice. But what are the baseline mTOR levels, right? The mice that where the big lifespan extensions come from are ad lib fed. So in general, they've overeaten. It would be nice to measure the effect size versus total calorie consumption. That work hasn't really been done yet. And in humans, of course, you would want to measure the effect size, in my opinion, stratified by BMI. Or ideally, actually measure the mTOR levels in any control group and experiment group. A lot of people are just jumping on board and taking rapamycin, including many people in the field who are experts. Well, let's pick a particular example that some people are excited about, sauna. There's lots of data from various different countries showing that sauna has all kinds of health benefits, heart benefits. The question is relative to what, right? How did, in the science experiments where sauna showed a benefit, what were the controls doing? I actually looked at a bunch of the papers, and some of them, there was like a 20-minute sauna, and then the controls just laid down on a table in a slightly warm room. And there's a lot of science suggesting that the benefit of sauna comes from mechanism of action and that overlaps with sweat. So where exercise and sauna might actually be doing some of the same things. So then you have to ask yourself, if you only have so much time in your week and you're already not exercising enough or not sleeping enough, is 20 minutes of sauna, adding that versus watching TV on the couch might be good, but is adding that versus creating less of an exercise deficit compared to what you should be doing, or creating less of a sleep deficit, which is better? That's not the way the studies are generally done. In general, there's a lot of things that affect rate of aging and health that fall into the category of hormesis, right? Slight stress that the body then adapts to, and if it doesn't kill you, it makes you stronger. And there's all kinds, right? Calorie restriction, which is one of the most reliable ways to increase lifespan, is a form of stress, dietary restriction in general, intermittent fasting. And then there's all kinds of cold, heat, various phytochemicals that are defense mechanisms of plants, are essentially mild poisons. Hypoxia, radiation in low doses has been thought to be hormetic. Even dehydration, which is referred to in some papers as intermittent drinking, which other people would think that means something else. Many stressors produce these beneficial adaptations, but dose is super important. And there's unfortunately no good work in this area that talks about interactions. So, the whole idea behind hormesis is that there's these U-shaped or J-shaped curves where there's a sweet spot of dose. And it's not clear, those U-shaped curves aren't mapped very well, and it's not clear how they interact, right? If your body gets stressed from one thing and you get simultaneously a different kind of stressor that also puts additional stress on it, I mean, it could be that each one of those stressors adjusts the position of the U-shaped curve for all the other stressors. But we don't actually know because we haven't detangled the science there. But that's an important thing to think about, right? If you're eating a lot of vegetables and having a low-calorie diet, maybe the cold or the heat or the radiation hormetic effect needs to be redosed for that. So keep that in mind. So care. So the previous slides were basically care needs to be. You need to be careful when interpreting these good benefits that you see in preclinical mouse data or in humans. Know who the controls are, but care is needed in interpreting negative studies too. So, for example, many vitamins trials fail because they gave the vitamins to people who already had plenty of that particular nutrient, or they didn't give enough of a dose to actually raise levels, or they gave something, but then it was easily available and the controls took it too. Some good papers on this. All right, in the Q&A, I'm going to wrap up soon, but in the Q&A, if we want to talk about Brian Johnson and Blueprint and epigenetic age clocks and I'm happy to talk about those things. Meditation's another one. The controls are, there's a lot of data on, for example, super stressed out CEOs who are super busy getting benefit from meditation, and it's less clear that people who sleep enough and things like that. So in general, aging interventions, you need to understand the mechanism of action, and because a lot of science is hitting these black boxes. What we really need in the field are better diagnostics that give us the steps before and after the mechanism of action, and so we can see if it's working as we thought it was. The field needs to look for interventions that have big effect sizes above and beyond optimizing lifestyle. And those are the things that are going to affect everybody. And the field needs heuristics for which interventions are going to combine well based on hitting different things instead of hitting the same things. That's pretty much it. I'm done. I'm happy to take Q&A as long as until the organizers say it's time for the Rejuvenation Technology presentation. Here are some topics that I could talk more about that I didn't have time to talk about in the slides.
Great, thanks. For those in the audience, if you have questions, go ahead and post them. My first question is, for mechanism of action, are any of these therapeutics on an FDA path, or is it a
Many
mixture?
of them, yeah. Plenty of them are on an FDA path. One of the columns in the company's table is the clinical, one of the columns in the company's table is the regulatory category. If it's I think it's called pharma for the ones that are on a normal clinical pathway, but there's several others, right? Some of the companies are just pursuing supplements, and some of the companies are pursuing veterinary applications first. So they're going after pets. And some of the companies are doing things that are totally different. They're, like smartphone apps. But the vast majority are on a standard pharma new drug discovery thing. And there's another column in the table which tells you the current clinical stage and has a link directly to the pipeline of the company. So you can, it'll say preclinical or phase 1, phase 2, or phase 3 trials, and you can click on that and it'll give you the full thing saying which indications they're going after and what their current stage is.
So a question from the audience is, how do you disambiguate between treatment and nature?
What do you mean by
You made the example of the vitamin where they already had enough of, say, vitamin B, and now we're giving them more vitamin B. How do you split out between what you're providing them and what is already naturally in the system?
Well, this is the problem. This is why some trials of vitamins or other nutrients will often fail. Vitamin D is the classic example where, you can't control how much sunlight everybody gets in the control group. And so there are some vitamin D trials where, for example, they started in winter and then they finish in summer, and presumably, and they've detected an increase in blood levels in both the control group and the experiment group.
Are there examples of therapeutics moving from the geroscience space into, say, the neurodegenerative or oncology space?
So at the moment, most of the therapeutics in the geroscience space have not made it through clinical trials. We're early in the field, and so there's many things in phase 1 and phase 2 and some in phase 3, and there haven't been a whole lot that have actually gotten approval. I consider things that focus on brain aging to be part of the space, even if they're neuro only, as long as they affect multiple different neurodegenerative diseases with the same mechanism of action. So Alzheimer's specific therapies I don't consider to be part of the space, but something that's a platform that has a therapeutic that's going to treat Alzheimer's and Huntington's and Parkinson's and ALS, that would be part of the space, within brain aging. And there are definitely lots of companies moving back and forth. There are definitely things that came out of geroscience where we discovered them in one area and they look like they affect neuro as well. And so then with companies pursuing that, or with a company spinning it out, or having a pharma partner.
So some people have better genes, according to the question in the chat box. Some men have heart attacks in their 50s, some are thin joggers, not fat smokers, and nature, not nurture ruled. So what do you think about the gene impact on this?
Most of the, genes absolutely affect rate of aging. They affect, there are genes that affect risk for each of the chronic diseases. And of course, centenarians who live above 100 and supercentenarians who live above 110 are known to have certain gene combinations that are one of the things that underlie their greater age. You can, for example, the biological children of supercentenarians tend to live longer than the general population. So we know there's something genetic involved. But that said, there's a lot of work that suggests that genes only only account for 4 or some single-digit percentage of the variation in lifespan, and that the majority of it is lifestyle. And regardless, no lifestyle and no genetic variation gets people past 120. So mostly there are genes that shorten life. There's no magical genes that cause us to live to 200. The biomedical research and development that's going on now, that's what's going to let people eventually live past the longest ever living human. And that's going to be regardless of what your genes are.
Great. And my last question, and then we'll have Glenn and John, if you want to get your slides ready. My last question is about telomerase. Can you tell us more about what that is? And heard about that's a key player here as well.
That is one of the categories I put on that list. 20 years ago, that was the only thing anybody really talked about in the aging field. And now the aging field has matured. So your DNA has these gene sequences on the ends that are that people usually analogize the little plastic caps that go on the ends of shoelace strings. Those DNA sequences help prevent the rest of the DNA from unraveling. And there are a certain sequence that gets repeated, and over time it gets shorter. Every time the cell divides, it gets shorter. When it gets too short, and then the DNA starts to unravel and the cells become senescent and all kinds of bad things happen. And with aging, they on average get shorter. And so some people try to extend them, including Rejuvenation Technologies, which we'll hear about. And so extending them has proven to be has some good data showing that it has benefit. And there's lots of the field has gone through some interesting transitions where there was some diagnostics where people were trying to, estimate people's age by looking at the lengths, and then that turned out not to actually be very predictive. And now they know that it's the critically short ones that seem, and that does seem predictive. So if you have very, very short telomeres, then that's clearly bad. And so we would want to extend those. And there's several companies working on this, and Rejuvenation Technologies, which, disclaimer, I am now an investor in, is one of them.
Great. Well, thank you for that. We'll switch over to Glenn Markov and John Ramunis of Regenerative Technologies. Karl, if you have a moment, please check out the questions in the chat box, and you're welcome to post some additional information there if you like. Glenn, John, go ahead.
Thank you, Karl. Can everyone see my slides?
Yes, we can.
Great. Yeah, so that was a great, really great overview by Karl. Thank you. And I think it sets us up really nicely to do a deeper dive in one of the hallmarks or pillars of aging, as they're called. So I'm one of the co-founders, Glenn Markov. I did my graduate studies at Stanford, and that's where John also was doing his research. And he was looking at ways to extend telomeres, and we found an extremely potent way of doing it. And that was exciting at the time, and still is, and we decided to launch the company around that. So this, as an intro to telomeres, which Karl did already a great job on, not only is it a major hallmark, and it is sort of the original hallmark of aging, if you will, and one of the ones that is most well studied, but one of the important things is considered one of the primary hallmarks, in that short telomeres give rise directly to many of the other kind of core hallmarks of aging, including senescence when telomeres become short, or genomic instability when chromosomes fuse with each other because of short telomeres, or inflammation as a result of deregulated signaling due to short telomeres. And of course, one of the most basic ones is the inability of cells to divide. And that is really what we're going after, looking at these age-related diseases where fundamentally there's a lack of regenerative capacity that results in cells unable to divide and repair tissues. And we know that's because telomeres short with age, and because short telomeres predispose to these diseases. And furthermore, if you have mutations in telomere maintenance genes and have prematurely short telomeres, you gain these diseases that it's really the telomeres that are driving this inability of your organs to recover. And so our goal as a company is to develop drugs that treat patients who have very short telomeres and bring their telomeres into the Goldilocks zone. And that will enable the loop at the end of their linear chromosomes to remain intact. The Goldilocks zone is quite wide space to land in. And so we believe we have a very good drug profile, in a sense where we can easily find and distinguish folks with these short telomeres. And with the potency of our drug, we have a relatively wide landing pad to make sure they're back into the normal range, which is about 10,000 bases in young adulthood, all the way down to about 5,000. And again, that's the average length. Importantly, it's the short telomeres that are the critical ones here. A summary of the technology is as follows. So there's a long outstanding effort to extend telomeres, and really was the advent of synthetic messenger RNA and John's first work that showed that with a single dose of this messenger RNA in human cells and culture, that an equivalent of a decade of telomere attrition could be reversed. And this is shown here with the histogram of telomere lengths at the bottom, where you can see the distribution of telomeres in the treated cells is quite shifted compared to the control cells in orange, which are human cells that have not received telomerase RNA, corresponding to about a 65% reduction in those short telomeres. And this is after 3 doses of the drug, which gave us about 1,000 base telomere extension, or about 20 years of telomere shortening reversed. And that was not just evident in the telomere lengths, but actually in how the cells looked like. They became less swollen, puffy. They had a reduction in senescent markers, so they were not as aged, if you will. And this messenger RNA, the way it functions is it's a molecule that the cells produce normally, so messenger RNA is very native to the cell. We produce it synthetically, but once the cell sees it, it knows what to do. It codes for telomerase protein, which is a limiting factor in cells to extend telomeres. And it's been honed by evolution to do just that. So we know these messenger RNA, very safe, non-toxic, codes for the protein, is transient, gets degraded, and the protein then goes and does the extension in the cell. And such, every dose, you get this pulse of telomerase. This is a real breakthrough in the field of telomere extension, as previously the attempts were largely using telomerase DNA, which is very effective, but it has the counter downside of delivering DNA, which then leads to genomic integration, long-term expression, and meaning you can't control the molecule. It's effectively turned on in some cells all the time. And that leads to immortalization. Other approaches have been tried to activate telomerase, which is very tightly regulated and have not been very effective. These companies are largely defunct or supplements, or in the case of danazol, it's a masculinizing hormone. The importance of telomerase as it relates to the cancer field is really the distinguishing factor that permanent telomerase can confer immortality while allowing cells to divide indefinitely. And that's what every single cancer cell has. It has some version of either permanent telomerase or another version of extending telomeres. In contrast, cells in the body naturally have transient telomerase, and as a result have longer telomeres. And so the questions around the synthetic telomerase messenger RNA are much more defined. And in fact, at our FDA meeting, there was no concerns around the telomerase approach specifically. And that's in part because we showed that telomerase activity is quite transient when we deliver it. And we've done studies in mice and others and have not seen an increase in tumor incidence. Upon launching the company, it was difficult to triage which indication to go after, as telomerase is, as short telomers affect many different tissues throughout the body during aging and give rise to many diseases. We ended up settling on a combination of diseases that have very strong unmet medical need, no current treatments, often fatal. Ones where the evidence is overwhelming that telomeres are the driver of the disease. And that includes evidence, again, in patients that have short telomeres because of a mutation, or they have inherited short telomeres from their parents and received these diseases at an earlier age. And those include pulmonary fibrosis, liver disease or liver cirrhosis, and then various forms of immunodeficiencies, including bone marrow failure. Also, importantly, we had to be able to deliver the messenger RNA into those tissues, and that's what we've been working on as a company. To build our pipeline of 3 different drug products, 2 of which I'll give some background today on. Our lead is a program to deliver the messenger RNA to the lung, and this can target many diseases. We're starting with pulmonary fibrosis, orphan disease designation available, and another one is liver disease, including compensated cirrhosis. And these are, as we'll go over, done a number of animal studies already. We're moving into the scale-up manufacturing work for the drug product before doing the IND-enabling toxicology to enable our first clinical trial. And we own the global rights for all of these programs. The drug itself is a mixture of the messenger RNA that does the active ingredient, if you will, with the lipids or excipients that are mixed to provide to both shield the RNA from external factors and also shepherd the RNA into its final destination. We've tested this in a number of studies, including this very novel LNP we developed to treat lung disease. And it targets the lung quite well, as you see in this photo here, where we've delivered RNA encoding luciferase, which causes the organ to glow if the RNA is delivered properly. And you can see the lung is glowing quite well, and majority of the signal is coming from the lung. And because this was a novel technology, we needed to test in multiple species, which we've done, both in animals, in mice, rabbits, and now non-human primates. And we've looked at within the lung and specific distribution of the transfected cells and saw that a great number of them were transfected in the alveolar compartment, we can see that over 95% of the cells are transfected at the upper dose. This graph here is a dose response of the drug. So the more we deliver, the more cells we can hit with a single dose. This is a summary of some of the animal data we've done so far. So we've looked at multiple different models of both pulmonary and hepatic fibrosis and disease. We see that treating mice after they've received an insult causing pulmonary fibrosis reduces that fibrosis by 60% or more, and results in a complete recovery of their forced vital capacity. And we see that you can see that in this photo here, where the yellow is the stain for the high density fibrotic regions. And this tissue section here is an entire lobe of the lung. So you can see how a good proportion of the lung in the control mice is yellow, and that yellow is reduced in the mice treated with telomerase lipid nanoparticles. We've seen similar evidence in the liver, where you can see again here about a 74% reduction of liver fibrosis. Again, this is the kind of the endpoint of the disease, but because telomeres are so fundamental, we see effects in many parameters of the tissue, including at the kind of microarchitecture level, looking at the things like alveolar density and diameter, and kind of parameters adjacent to the disease, including inflammation. In both these liver and lung models, we see a big reduction in inflammation. Looking at the safety profile, we've seen that our drugs have very rapid clearance of both the lipid and the RNA, which is important for safety. So it's a hit and run mechanism. We deliver the LNP, it goes to the site, and then it's cleared quite rapidly. We've had a board certified pathologist examine the tissue at a microscopic level and see no moderate or severe findings in any of the 6 tissue panels shown here, including no findings at all in the lung, which we were primary kind of organ of concern for our lung LNP. And of course, for the liver LNP would be the liver. We've looked at a number of other kind of chemistry panels and hematology panels, as well as plasma cytokines for immunogenicity, to make sure that after multiple doses, the baseline cytokine levels are restored. And that is the case here. You can see that on the right, looking at 6 different markers. So our path is towards the FDA. We are looking at manufacturing and toxicology studies in the next year to year and a half, whereby we can begin our phase 1 clinical trial. And that's for the pulmonary program, and the hepatic program is just maybe 6 months behind that. And our goal is to have ultimately data, phase 1 data in patients suggesting that our drug is safe, we have a dosing regimen and a dosing frequency that we're comfortable with, and some movement and biomarkers suggesting that it's working already. And companies that have had that type of data are typically valued in the 1 to 5 billion range. So in terms of our current funding round, we were looking at a $2 million seed extension round, of which 1.6 million we've already received. It's led by Merchant Ventures here. And we also have a number of grants that are starting or have started already, including a large one from the California Institute of Regenerative Medicine for our pulmonary program, and the National Institute of Health for our hepatic program. And these funds will be used for our drug product manufacturing and our toxicology studies, as well as patent protection, because the grants don't cover IP spending. On a broader sense, we have a very good track record of venture funding since our founding in 2018, where we were first funded by Y Combinator, and we've raised over 10 million in investor capital today. We'll be targeting a Series A term sheet the next 6 to 9 months, looking at pushing us into clinical trials. And these are some of the investors so far on the left here. We've also been engaging with multiple large pharma partners to build those relationships and to make sure that ultimately we know, get that feedback early to understand what they're looking for, what makes sense to them, and ultimately we're having those options to engage for possible deal, most likely after a phase 1, since that's when the program is most de-risked and the valuations are high. We have a growing IP portfolio. I'm displaying some of our core patent families here, including the IP around the use of telomerase messenger RNA for telomere extension, as well as the use of telomerase messenger RNA for treating liver disease, and our novel lung composition lipid nanoparticle, as well as, of course, we have exclusivity rights for any biologics in US and Europe. And I'll note that the telomerase messenger RNA patent was an exclusive license from Stanford, whereas the others were filed from the company. We have a really amazing and rapidly growing team, including John and myself. Susanna is our director of both our project management side and the scientific research, and has experience in a multi-billion-dollar GMP production environment. We recently have made kind of verbal agreement with Dan Chambers, shown here, to be our acting chief medical officer for our phase 1 trial. He runs one of the largest clinical sites in the world for recruiting patients for pulmonary fibrosis, and is really a clinical champion in the field for this hypothesis. And that also includes Paul Wolters and Christine Garcia, who have published many papers and dedicated their line of research to demonstrating the importance of short telomeres in pulmonary disease. And we have distinguished folks and advisors on various narrow verticals of the drug development process, specifically for messenger RNA lipid nanoparticles, which is a relatively new field. And we feel like we have the best people for each of those verticals, such as toxicology of the drugs, bioanalytical development, and manufacturing of the lipid nanoparticle. So that's the conclusion. Happy to take questions.
Sure. In the chat box, the first one is, do you think avoiding cellular senescence is the primary benefit of extending telomerase?
I think the senescence is a very important part. We do see decrease in senescence in our studies. I also think that by enabling the block of senescence from occurring, we're simultaneously allowing the cells to divide more. And that really, I think, it fundamentally enables regeneration. So even if you're just simply clearing senescent cells, for example, if you're not enabling the remaining cells to divide more, you're only going to have more senescence in the end. And so you're not really, at the fundamental level, you're clearing the junk out, but you're not kind of repairing what the problem at hand. So I think we're doing that and more.
Are there any other notable benefits that you think are mediated by decreased numbers of cells becoming senescent?
Absolutely. There's, I think, great benefits around the signaling that happens when the cells are no longer senescent. And in our case, because we're extending telomeres, we're also improving the genomic stability of the cells, so the chromosomes are no longer fusing with each other. So that can, of course, then actually prevent, in some cases, oncogenic mutations.
Great. And Karl, can you tell us about, you're an investor in the group. Why did you invest in this one?
Well, he went over it pretty well in the thing. I mean, any comprehensive solution to aging is going to need to extend telomeres, and there's a long history of companies working on this and not really going anywhere. And it just seems like it's hard to do with small molecules because of all kinds of off-target effects. And I'm not really comfortable with the full-on gene therapy, permanent approach. So the RNA approach just seems perfect for the hit and run, as he said, and that you can just so precisely time the dosing, and of course, combined with now after COVID, the RNA is almost a fast track from a regulatory perspective. People are very much more comfortable with it, so it just seems like it's the company with the sweet spot way to extend telomeres, and so that I think it's great, and it's of course great that they got all this grant money. So I think they're well placed to have runway until the markets come back.
And what do you think has been their progress in the past couple of years? Is it on track, ahead of schedule, behind schedule? What do you think it looks like there?
I mean, as far as I can tell, they're hitting milestones and things. I've only been an investor personally myself since a tiny bit earlier in 2023. So I haven't followed along as an investor for very long yet.
Okay, no, that sounds great. Are there any other questions from the audience? Love to have them in there as well. Here's one. Can you give any insights into the NHP data?
Yep, absolutely. So the data is sort of still coming in. We're still processing those tissues, but I can give you guys sort of a view here. So we've looked at non-human primate kind of organ by organ where our drug is being expressed, and we see again as kind of confirming that primarily we're looking at the lung as the primary site of delivery for our novel technology. And so we're doing a number of kind of follow-on studies examining the rate of drug clearance and safety, but importantly, there were no clinical signs observed even at our higher dose. We can see here sort of if you just distribute the tissues by size that primarily it's lung focused on both the high dose and the low dose. And so I think that bodes really well for our clinical both our clinical trials, but also our actual toxicology studies, which will be done in the non-human primates.
Great. And where do you think is the initial exit going to be? Is it at FDA approval, or do you think it'll happen sooner?
Yeah, I think there is a big, so apologies, I'm going to jump to this slide here. I think there's a big interest in pharma companies because this is a potentially $20 billion a year type of opportunity. Upon approval, there's a big appetite to take that on well in advance as soon as there is some clinical data that enables the pharma to say, hey, this is something that we could do. Or in contrast, in the past years, and typically the public markets are very accepting of a new company going public that has this, some type of clinical data in a new technology like this. And so we've talked to a couple folks who are currently CFOs at public companies, and that's what they believe as well. So I think those are kind of our two options that we'll see, and I think that will happen much earlier than the final drug approval.
Is all pharma interested in this space and they have something going, or is it just a small cottage industry of pharma that are going to go after it?
Yeah, I would say for the pulmonary fibrosis and increasingly chronic diseases, as we see the biggest blockbusters this year are things like the GLP-1 antagonists from Nordic and Lilly. So those guys are bringing in tens of billions for these age-related disease with a mechanism that is potentially mediating some amount of nutrient sensing and aging pathway. So I would say of the top, 20 or so or 10 pharmas, roughly a third of them are interested in the space just based on the conversations we've had.
Great. Good chat going on in the chat box. Jordan, do you have any thoughts about this so far you want to share with us? We'll see if he gives us a response there. I know it's the last minute here, but we're near the end of our time. We'll give people just a minute more if you want to put a thought or a question into the chat box. And wanted to see, okay, go ahead and Glenn, give us some final words there on next steps here for you guys.
Yeah, so I think we're well placed to kind of continue our work as we've described around our scale up of our drug manufacturing and some of our toxicology studies. And for the seed extension round, we'd love to have any last minute participation as we'll be turning our focus kind of mid-September to talking to some of the bigger Series A venture funds and seeing what their interest is.
Well, great. Well, Jordan put in there that he was really happy with all the data and the timeline. So looks like you're on track with that as well. And then all the non-dilutive funding is a really good sign as well. That really validates the technology if you get that much money for it. So looks like he has some positives there also. If there are no other questions, guys, we'll go ahead and wrap up for today. Anything else from your side, Karl?
No, just I encourage everybody to follow along on AgingBiotech.info. There are email addresses to contact there, and it's a great space. You should pay attention to it.
Okay. Thanks, Glenn. Thanks, Karl.
Guys.
Thanks, John, for coming out today. With that, we're going to close it up for today, guys. We will send you the recording and the chat box so you can check it out there, and we can give you more details about Rejuvenation Technologies with the deck as well. But with that, we'll go and wrap it up and see you guys next time.